Sleep and dreams are governed by complex neurophysiological processes that involve distinct patterns of brain activity and chemical regulation, ensuring both physical restoration and cognitive emotional balance. Core brain structures implicated in the regulation of sleep include the hypothalamus, thalamus, brainstem, and the prefrontal cortex, while dream generation is strongly linked to limbic and associative networks. In healthy individuals, the hypothalamus serves as the primary regulator of circadian rhythms through the suprachiasmatic nucleus, SCN, which synchronizes the sleep-wake cycle with environmental cues such as light and darkness. The brainstem, particularly the pons, coordinates transitions between different sleep stages by regulating neurotransmitters like acetylcholine, serotonin, and norepinephrine. During rapid eye movement, REM sleep, acetylcholine activity intensifies while monoamine neurotransmitters decrease, creating the unique conditions in which vivid dreaming emerges. Dream activity is associated with heightened activity in the amygdala and hippocampus, regions central to emotional processing and memory consolidation. This explains why dreams often integrate intense feelings, past experiences, and fragmented memories. At the same time, there is decreased activity in the dorsolateral prefrontal cortex, the region responsible for logic and self-monitoring. This reduction in executive control contributes to the illogical, surreal, and often bizarre nature of dream narratives. On a neurochemical level, GABA and galanin promote deep, slow-wave sleep by inhibiting arousal systems, allowing the body to restore energy, strengthen the immune system, and regulate hormonal balance. In contrast, REM sleep involves dynamic neurochemical shifts that support synaptic plasticity, learning, and emotional regulation. The alternating cycles of non-REM and REM stages across the night reflect a coordinated interplay between restoration and cognitive emotional processing. Sleep disturbances, whether caused by stress, trauma, or neurological conditions, disrupt these finely tuned mechanisms. Reduced slow-wave sleep can impair physical recovery, while fragmented REM sleep interferes with memory consolidation and emotional regulation. For instance, individuals exposed to chronic stress may experience hyperactivation of the amygdala during sleep, leading to more intense or disturbing dreams. Similarly, a lack of deep sleep weakens prefrontal regulation over limbic structures, intensifying vulnerability to mood disorders and cognitive difficulties. Developmentally, sleep architecture changes across the lifespan. Children experience greater proportions of deep sleep, supporting rapid brain growth, while older adults often exhibit reduced slow-wave and REM activity, contributing to memory decline and emotional instability. Environmental factors such as irregular schedules, artificial light exposure, or chronic stress can further desynchronize circadian rhythms, amplifying risks for insomnia, mood instability, and impaired concentration. By examining sleep patterns and dream content, researchers can better understand how the brain organizes memories, regulates emotions, and adapts to life experiences. Dreams reflect the brain's attempt to integrate past experiences with current emotional states, while sleep itself functions as the foundation for mental clarity, physical resilience, and adaptive coping in daily life.